Okay, so I'll start the class now. So, hello everyone. Today is the second problem solving session for the course Overview and Integration of Cellular Metabolism. So, in the previous class, we discussed glycolysis in detail, and then we started with the TCA cycle, right? And we discussed about how enzymes are regulated by allosteric mechanisms. It can be allosteric positive regulation or a negative regulation. And then we discussed all the steps of glycolysis, and uh, then we moved on to TCA. So today in this session we will discuss more about TCA, then the regulation of glycolysis, TCA, and we'll discuss about a new cycle, that is neo-glucogenesis or gluconeogenesis, which is basically a reverse cycle of TCA, reverse cycle of glycolysis. Then we will move on to uh, the regulation of gluconeogenesis, and finally HMP shunt pathway. Okay, so you must have gone through the lecture series of this week's second week's video. So if you have any doubt in any of the topic, feel free to ask. I'll solve some questions uh, related to the topics of this week, and like you can answer those questions, and I'll explain the concepts along with the questions. And if you have any doubt in any of the questions, or if you have any doubt in any topic in general, you can ask me. Okay, so I'll start with the first question. First question is, which of the following reaction does not contribute to amphibolic nature of TCA cycle? Option A, pyruvate to malate by malic enzyme reaction. Option B, succinyl coenzyme A to purines. Option C, alpha ketoglutarate to glutamate. Option D, citrate to fatty acid. And option E, succinyl coenzyme A to porphyrins or heme. Which of the following reaction does not contribute to the amphibolic nature of TCA cycle? So if you know the answer, you can write it in the chat box. I see Meenakshi has written option B, that is succinyl coenzyme A to purines. Okay, so both of you are agreeing to option B. That is the correct answer. So, do you know what amphibolic nature means? What does amphibolic nature mean? Both anabolic and catabolic. Yes, that is exactly correct. So basically, TCA cycle has both anabolic and catabolic nature. Uh, Meaning, catabolic means breakdown, just in very simple words. Catabolic means breakdown, and anabolic means synthesis. Okay, so TCA is obviously involved in breakdown. We saw in the previous class that uh, in glycolysis, we had glucose breaking down into pyruvate, then pyruvate was getting converted into acetyl coenzyme A, and then that acetyl coenzyme A was entering the TCA cycle. And ultimately, the products of TCA cycle, all the electron carriers, NADH and FADH2, all those molecules will enter the electron transport chain or the oxidative phosphorylation pathway, where more and more ATP will be generated. So at the end, everything is broken down to generate energy. That is the catabolic nature of TCA. Anabolic nature means synthesis. What all is synthesized in TCA cycle? So you know, TCA, all the intermediates of a TCA cycle are actually precursors of different biomolecules. So here you can see, so this is the TCA cycle we have, where acetyl coenzyme A is entering the pathway, and it is getting converted into citrate. Then alpha ketoglutarate, succinyl coenzyme A, malate, oxaloacetic, all these are intermediates of citric acid cycle. Now, here you can see citrate can be a precursor of fatty acids and sterols. Alpha ketoglutarate is a precursor of glutamate, which in turn is involved in the synthesis of other amino acids like glutamine, proline, and arginine. Also, glutamate is also involved in the synthesis of purines. Succinyl coenzyme A similarly is the precursor of porphyrins or heme. Then we have pyruvate getting converted to malate, which is another intermediate of TCA cycle. Then oxaloacetate is a precursor of phosphoenol pyruvate. We'll see this path uh, in the subsequent questions also. So oxaloacetate can get converted into PEP, phosphoenol pyruvate. If you remember, phosphoenol pyruvate is a part of glycolysis, the second last step, the last step of glycolysis, where the second last compound is phosphoenol pyruvate, which gets converted into pyruvate. So oxaloacetate can actually get converted into phosphoenol pyruvate directly, which can either go back to glucose by gluconeogenesis, which we'll discuss in detail in the subsequent questions, or phosphoenol pyruvate can also be a precursor to certain amino acids like serine, glycine, cysteine, phenylalanine, tyrosine, and tryptophan. Okay. And oxaloacetate can also be precursor to aspartate, asparagine, which are other amino acids, and these amino acids can be precursors of pyrimidines. Okay, so how glutamate is a precursor of purines, aspartate is a precursor of pyrimidines. Okay, and purines and pyrimidines, you know where it is required. It is required for the nucleic acid synthesis, right? Your DNA and RNA are comprised of purines and pyrimidines. Okay, so what all options did we have? We had pyruvate to malate by Maillard enzyme reaction, that we have here. Then we have succinyl coenzyme to purines. That is the wrong option, because succinyl coenzyme is a precursor of porphyrins and hemes. Purines, glutamate is the precursor of purine. Okay. Then we have alpha ketoglutarate to glutamate, that is correct. Citrate to fatty acids, that's also correct. And then succinyl coenzyme to porphyrins or hemes. Okay, so both of you answered it correctly. That succinyl coenzyme A to purine is uh, not the reaction that shows amphibolic nature of TCA, right? Okay. Moving on to the next question then. Anaplerosis refers to the replenishment of intermediates in the TCA cycle. Which of the following is an anaplerotic reaction? Anaplerosis refers to the replenishment of intermediates in a TCA cycle. Which of the following is an anaplerotic reaction? Option A, conversion of oxaloacetate to malate. Option B, conversion of citrate to isocitrate. Option C, conversion of alpha ketoglutarate to succinyl coenzyme A. And option D, conversion of pyruvate to oxaloacetate. Which of the following is an anaplerotic reaction for TCA cycle? Okay, I see one answer in the chat box. Meenakshi has written option D, that is conversion of pyruvate to oxaloacetate. Okay. Subhaprata, do you also agree with this answer? Okay. So both of you are saying option D, that is conversion of pyruvate to oxaloacetate, as the correct answer. So that is the correct answer. So anaplerosis basically refers to the replenishment of the intermediates. Uh, Here you can see, I told you in the previous slide that the intermediates of the citric acid cycle or the TCA cycle are precursors to different other biosynthetic pathways. Okay, so now if the citrate is going to go for fatty acid synthesis, or alpha ketoglutarate is going to do glutamate synthesis, then there will be a need to replenish these citrate or alpha ketoglutarate or malate or oxaloacetate in the cycle, right? So you see the cycle starts with oxaloacetate, uh, where the acetyl coenzyme enters the TCA cycle, and along with oxaloacetate it forms citrate. Okay, so the first anaplerotic reaction for TCA cycle is the conversion of pyruvate to oxaloacetate, where pyruvate can directly be converted into oxaloacetate in order to replenish the oxaloacetate that is required in the TCA cycle. And where does this reaction occur? It occurs in the liver and kidney, mainly in the liver and kidney. Okay, and which enzyme catalyzes this reaction? Pyruvate carboxylase. So pyruvate is a 6 carbon compound. uh, Pyruvate is a 3 carbon compound, so you need an addition of extra carbon to form a 4 carbon compound, that is an oxaloacetate. Okay, so this is 3 carbons and 1 carbon is coming from the bicarbonate ion, and we are getting a 4 carbon compound, that is oxaloacetate. Okay, so pyruvate carboxylase catalyzes the reaction from pyruvate to oxaloacetate, and it is an energy consuming reaction. It requires ATP. Okay, so one ATP molecule is converted into ADP and PI. So one ATP molecule is invested in this reaction. So it's an energy consuming reaction, but it is required by the by our system to replenish the oxaloacetate that has gone for other biosynthetic pathways. Okay, similarly, there's another anaplerotic reaction that replenishes oxaloacetate, that is from phosphoenol pyruvate. So phosphoenol pyruvate by phosphoenol pyruvate carboxykinase and phosphoenol pyruvate carboxylase. Both of the enzymes can do this conversion of phosphoenol pyruvate to oxaloacetate. So in heart and skeletal muscle, phosphoenol pyruvate carboxykinase is present, and since it's a kinase, obviously a phosphate transfer reaction is involved. So here GDP uh, is converted into GTP. So a phosphate is uh, transferred from this phosphoenol pyruvate. So phosphoenol pyruvate has a phosphate, right? So this phosphate is taken up by this GDP to become GTP. This GTP can further uh, go and get synthesized into ATP. So ultimately, we will get one energy molecule. Okay, and again, I told you pyruvate is a 3 carbon, so 1 carbon dioxide is required. So that's it's a carboxy kinase. So carboxylase, wherever it is written, that means a uh, carbon dioxide or a bicarbonate is added. Okay, carboxy kinase means a carbon dioxide is added plus a phosphate molecule is transferred. Okay, then you have the other reaction where again phosphoenol pyruvate is converted into oxaloacetate. This is catalyzed by the enzyme phosphoenol pyruvate carboxylase, which is present in uh, higher plants, yeast, and bacteria. And in this reaction, there's no transfer of uh, phosphate group from one molecule to another. The phosphate is just released as an inorganic phosphate moiety. Like here, the phosphate was transferred to GDP. That's why a kinase was involved. Here, only carboxylase is involved as CO2 is added, and phosphate group is just released as inorganic phosphate. Okay. Another anaplerotic reaction that replenishes malate, which is another intermediate of TCA. So pyruvate is converted into malate by malic enzyme. So by malic enzyme, pyruvate can be converted into malate again by addition of 1 carbon, because malate is also a 4 carbon compound. And in this reaction, one NADH molecule or NADPH molecule is utilized. Okay. So it is mostly it is widely distributed in eukaryotes and bacteria. So it can occur at multiple locations. Okay. So these are four anaplerotic reactions in TCA that can replenish the intermediates of the TCA cycle. So once oxaloacetate is replenished, all the other things can easily be replenished by combining with acetyl coenzyme A. Or if malate is replenished, malate can become oxaloacetate, and then other things can be replenished easily. Okay. So that's how the intermediates of TCA cycle is being replenished. Any doubt in this? If you have any doubt, you can write it in the chat box. Okay. Moving on to the next question. Which of the following reaction is irreversible in glycolysis? Which of the following reaction is irreversible in glycolysis? Option A, glucose 6 phosphate to fructose 6 phosphate. Option B, phosphoenol pyruvate to pyruvate. Option C, 3 phosphoglycerate to 2 phosphoglycerate. And option D, glyceraldehyde 3-phosphate to 1,3-biphosphoglycerate or bisphosphoglycerate. Which of the following reaction is irreversible in glycolysis? Glucose 6-phosphate to fructose 6-phosphate, phosphoenol pyruvate to pyruvate, 3-phosphoglycerate to 2-phosphoglycerate, and uh, glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. I see Meenakshi has written option B, that is phosphoenol pyruvate to pyruvate. Okay. Is that the correct answer? Okay, so both of you are agreeing to option B, that is PP to pyruvate. That is the correct answer. Just a second. So phosphoenol pyruvate to pyruvate, that is the correct answer. So in glycolysis, we discussed in the previous session also that we have 10 steps in glycolysis, right? So out of these 10 steps, we have 3 steps that are irreversible. Okay. So you can see glucose to glucose 6 phosphate. The first step, that is catalyzed by hexokinase. This is irreversible. So for the reverse reaction, glucose 6 phosphate to glucose, we need another enzyme. Similarly, fructose 6 phosphate to fructose 1,6 bisphosphate, which is catalyzed by phosphofructokinase PFK1. This is also irreversible, and for the reverse reaction, we need another enzyme. And third is phosphoenol pyruvate to pyruvate, which is catalyzed by pyruvate kinase, and this is also irreversible. Okay, so if you see, mostly all the 3 steps where phosphorylation is happening, those steps are irreversible. So if you need a trick to remember it, you can remember it like that. So 3 steps where phosphorylation is happening, those steps are irreversible. Okay. So why we are talking about reverse reaction is, next we will learn about gluconeogenesis, which is just the opposite cycle of glycolysis. So in gluconeogenesis, for the other steps, we know that the same enzyme can catalyze the reverse reaction, but for the steps where we need another enzyme, so for for the step where we have to go from fructose 1,6 bisphosphate to fructose 6 bisphosphate, we need another enzyme. Similarly, from glucose 6 phosphate to glucose, we need another enzyme. Pyruvate to PEP, we need another enzyme. Okay, so we'll see that in the next question. Any doubt in this, in the previous question? Okay. The enzyme responsible for the conversion of pyruvate to phosphoenol pyruvate in gluconeogenesis are the enzyme that are responsible for conversion of pyruvate to phosphoenol pyruvate in gluconeogenesis are option A, pyruvate carboxylase, option B, phosphoenol pyruvate carboxykinase, and option C, fructose 1,6 bisphosphatase, and option D, glucose 6 phosphatase. Enzymes responsible for the conversion of pyruvate to phosphoenol pyruvate in gluconeogenesis. Option A, pyruvate carboxylase. Option B, phosphoenol pyruvate carboxykinase. Option C, fructose 1,6 bisphosphatase, and option D, glucose 6 phosphatase. So multiple options could be correct. So answer accordingly. So both of you are saying option A and option B. That is pyruvate carboxylase and phosphoenol pyruvate carboxykinase. Okay. So those two are the right answer. Option A and option B, pyruvate carboxylase and phosphoenol pyruvate carboxykinase. So I told you there are three steps in glycolysis which were irreversible. So here you can see we have the glycolysis in one direction and gluconeogenesis in the other direction, in the reverse direction. So here you can see uh, the blue side is basically glycolysis and the red side is gluconeogenesis. Okay, so from glucose to glucose 6 phosphate, we know in glycolysis hexokinase is involved. So instead of hexokinase, we have glucose 6 phosphatase that will remove the phosphate. Now phosphatase are the group of enzyme that can remove phosphates. Okay, so glucose 6 for glucose 6 phosphate to get converted back to glucose, we need a phosphatase enzyme to remove the phosphate, to remove the 6 carbon phosphate. Okay, so for that we have glucose 6 phosphatase. Similarly, from fructose 1,6 bisphosphate, we want to make fructose 6 phosphate. That means we want to remove one phosphate. So the enzyme that will be required is fructose bisphosphatase. Okay, for addition of phosphate or for transfer of phosphate group from one molecule to another, we use the enzyme kinase. Okay, for removal of phosphate, we use the enzyme phosphatase. Phosphatase generally releases an inorganic phosphate from the substrate molecule. Okay, then we have the third irreversible reaction, that was phosphoenol pyruvate to pyruvate, as we saw in the previous question. The enzyme responsible for the conversion of PEP to pyruvate was pyruvate kinase. So for pyruvate to go back to phosphoenol pyruvate, we have two enzymes. It's a two-step process. So pyruvate cannot get directly converted into phosphoenol pyruvate. So it goes through oxaloacetate, and pyruvate to oxaloacetate, I told you this is an anaplerotic reaction. It replenishes the oxaloacetate in the TCA cycle. So pyruvate carboxylase is the enzyme which uses one ATP molecule and a carbon dioxide or a bicarbonate molecule to get converted into oxaloacetate. And then from oxaloacetate, we have phosphoenol pyruvate carboxykinase. Again, a carboxykinase, that means a carbon dioxide is involved and a phosphate group transfer is involved. Okay, so a phosphate group from GTP is taken. So GTP is converted to GDP, and that phosphate group goes to pyruvate, making it phosphoenol pyruvate, and the carbon dioxide gets eliminated from the oxaloacetate. Okay, so oxaloacetate is a 4 carbon molecule. Okay, so that's how the whole reaction is being regulated. And we'll see where this reaction occurs. So this reaction mainly, so pyruvate is present in the cytosol, right? Pyruvate is a glycolysis product. It is present in the cytosol. Glycolysis occurs in the cytosol, so it is present in the cytosol. Now, from and we know oxaloacetate, where do we have oxaloacetate? We have it in mitochondria. TCA cycle is occurring, right? So pyruvate is transferred into the mitochondria, where pyruvate carboxylase is present. Pyruvate carboxylase converts pyruvate into oxaloacetate. Then that oxaloacetate now cannot be carried out of the mitochondria. So mitochondrial membrane do not have a transporter that can transport oxaloacetate directly. Okay, so that's why oxaloacetate is converted into malate by malate dehydrogenase. So it's a mitochondrial malate dehydrogenase. Basically, the enzyme is present inside the mitochondria. Then this malate is then transported out into the cytosol, and it is again converted back to oxaloacetate by malate dehydrogenase, now this time cytosolic malate dehydrogenase. And this oxaloacetate can be converted into phosphoenol pyruvate by phosphoenol pyruvate carboxykinase. Okay, and in this whole process, that for oxaloacetate to get converted into malate, one NADH molecule is being used up, and when malate is converted into oxaloacetate, that NADH molecule is released back into the cytosol. Okay, there is another pathway that follows where lactate is the starting molecule. So if pyruvate is the starting molecule, then in that case this pathway is followed, that is given on the left hand side. But in case lactate is the starting molecule, high amount of lactate has accumulated in the muscle. In that case, lactate is again converted back to pyruvate by lactate dehydrogenase. This lactate dehydrogenase, when it converts lactate to pyruvate, it will release one NADH in the cytosol. This pyruvate will then be carried into the mitochondria through the membrane, and then pyruvate can be converted into oxaloacetate by pyruvate carboxylase, just like the previous step. And this oxaloacetate can directly get converted into converted into phosphoenol pyruvate by mitochondrial PEP carboxykinase. Now, obviously, you will ask like why here this oxaloacetate is not converted directly by this enzyme. The reason this pathway is favored in case when pyruvate is directly converted into phosphoenol pyruvate is because we need one NADH to be released in the cytosol. Since in case of lactate, we are getting that NADH directly from lactate dehydrogenase, we don't need to go through this whole pathway. Okay, but in this case, if oxaloacetate would have got converted into phosphoenol pyruvate directly with the PEP carboxykinase, the level of NADH will be low in the cytosol. So NADH levels are generally low in the cytosol and high in the mitochondria. Okay, but to trigger gluconeogenesis, to trigger this reverse reaction, we need high amount of NADH, because more amount of NADH means we already have energy currencies. Okay, so now we need to do a reverse reaction to make these to use up these energy currencies to make more and more glucose, so that our body can again trigger those glucose into glycolytic pathways. Okay, So it's basically a back and forth mechanism where glu- whatever you are eating ultimately breaks down to glucose, and then those glucose molecules are uh, metabolized through glycolysis, converted into pyruvate, which enters the TCA cycle through acetyl coenzyme, and then finally we get ATP. But in case where let's say you are fasting, okay, so in that case now your body still has to function, your body, your brain is still functioning. To for any organ to function, it needs energy. In that case, what will it do? It will start breaking down the other metabolites that are not carbohydrate sources, because since you are fasting, your body has accumulated other metabolites as well, and they will convert those to glucose, so that that glucose can then be metabolized. Then again glycolysis can be triggered and ATP generation can occur. Okay, so that's why gluconeogenesis is an important pathway. Do you understand this? I hope that's clear to everybody. I'll move on to the next question. Following are the substrates for gluconeogenesis except option A, glucogenic amino acids, option B, glycerol, option C, propionyl coenzyme A, option D, lactate, option E, saturated fatty acids, and option F, pyruvate. Following are the substrates of gluconeogenesis except glucogenic amino acids, glycerol, propionyl coenzyme A, lactate, saturated fatty acids, and pyruvate. So both of you have written the option E, that is saturated fatty acids. So all of the other options are substrates of gluconeogenesis. Only saturated fatty acid is uh, not a substrate of gluconeogenesis. So option E is the correct answer. So here are the different substrates of gluconeogenesis. So as I've mentioned in the options, glucogenic amino acids, glycerol, propionyl coenzyme A, lactate, and pyruvate. All of them can enter the gluconeogenic pathway. So I told you gluconeogenesis is nothing but the formation of glucose. So glycolysis was breakdown of glucose. In this case, we are making more glucose. Okay, so if lactate is present or pyruvate is present, it can directly uh, enter the citric acid cycle. How? By the formation of oxaloacetate. We saw in the previous slide, pyruvate or lactate can get converted into pyruvate, and then that pyruvate can get converted into oxaloacetate. That oxaloacetate can then be converted into phosphoenol pyruvate, right? We saw that in the previous question. Similarly, there are certain glucogenic amino acids. So here's a list of glucogenic amino acids that can enter the TCA cycle at different points. So there are certain amino acids like alanine, cysteine, glycine, serine, threonine, and tryptophan. All of these can enter the citric acid cycle by by getting converted into pyruvate. So pyruvate won't enter the citric acid cycle. Pyruvate will then get converted into oxaloacetate, and that can enter the citric acid cycle, and that can ultimately generate phosphoenol pyruvate. Similarly, isoleucine, methionine, threonine, valine, this can get converted into succinyl coenzyme A, and that succinyl coenzyme A can enter the citric acid cycle. And that succinyl coenzyme A, once it enters the TCA, it will obviously get converted into oxaloacetate, and then oxaloacetate can go into PEP. Okay. Similarly, we have phenylalanine and tyrosine that can get converted into fumarate, which is another intermediate of TCA cycle, which can get converted into oxaloacetate and uh, ultimately form PEP. Then we have arginine, glutamate, glutamine, histidine, and proline, which can get converted into alpha ketoglutarate, and that can ultimately get converted into oxaloacetate and phosphoenol pyruvate. And we have asparagine and aspartate that can get converted into oxaloacetate. So these are all glucogenic amino acids that can enter the TCA cycle at different time, at different intermediate stages. Okay. Then we have glycerol or triacyl glycerols. So basically, fatty acids are broken down into triacyl glycerol, and that triacyl glycerol can now get converted into glycerol and enter the gluconeogenesis pathway by getting converted into glucose 6 phosphate. So glycerol will get converted into glucose 6 phosphate, and glucose 6 phosphate can get converted into glucose, uh, as we saw in the previous slide. Then we have CO2 fixation, which is not present in animals. It is only present in plants and photosynthetic organisms. So animals cannot do CO2 fixation, but plants can directly fix CO2 into 3-phosphoglycerate, and that 3-phosphoglycerate can get converted into glucose 6-phosphate. It can basically enter the gluconeogenesis pathway. So glucogenic amino acids I explained, glycerol. Propionyl coenzyme A can also enter the gluconeogenesis pathway through formation of acetyl coenzyme A, which can then go in the reverse direction. Okay. Then we have lactate, I explained, and pyruvate. So saturated fatty acids are not a substrate of gluconeogenesis. Saturated fatty acids are directly broken down into acetyl coenzyme A. Okay. Is this clear? Till now, if you have any doubt, please let me know. So moving on to the next question then. Which hormone stimulates gluconeogenesis and inhibits glycolysis? Which hormone stimulates gluconeogenesis and inhibits glycolysis? Option A, insulin. Option B, glucagon. Option C, epinephrine. Option D, cortisol. And option E, norepinephrine. Which hormone stimulates gluconeogenesis and inhibits glycolysis? Option A is insulin, option B is glucagon, option C is epinephrine, option D is cortisol, and option E is norepinephrine. So both of you have written the answer glucagon, option B. That is the correct answer. So, if you remember in the previous session, we discussed about insulin, how insulin can modulate the glucose levels. Okay, and why insulin was important in controlling diabetes, right? So, we had studied that in the presence of insulin, the blood glucose level is basically controlled. So, if more and more insulin is present, more and more glucose will be metabolized into glycolysis, metabolized by glycolysis pathway. Similarly, there's another hormone called glucagon that stimulates gluconeogenesis. So, if so insulin and glucagon, they both work simultaneously. If insulin levels are high, then glycolysis will be preferred and gluconeogenesis will be inhibited. If glucagon level is high, then glycolysis will be inhibited and gluconeogenesis will be triggered. So basically these two hormones are regulatory hormones, and they basically regulate the levels of glucose in our body. So once when our body has sufficient amount of glucose, will be secreted out of pancreas, and then insulin will trigger the uh, glucose transport and glycolytic pathway. Whereas when our body is having glucose deficiency and it requires more and more glucose or more energy, in that case, what it will do, glucagon would be the hormone that will be secreted, and then that hormone will trigger the gluconeogenetic pathway and it will inhibit the glycolysis. So it will stop the utilization of glucose and it will start making more and more glucose. Okay, so that's how the uh, regulation between gluconeogenesis and glycolysis happens by these two hormones, insulin and glucagon. Okay, so more insulin means more glycolysis, less gluconeogenesis. More glucagon means more gluconeogenesis and less glycolysis. Is that clear? Okay. Okay, moving on to the next question. The reciprocal regulation of glycolysis and gluconeogenesis is mainly controlled by the interplay of two key enzymes. These enzymes are option A, hexokinase and glucose 6 phosphatase, option B, phosphofructokinase 1, that is PFK1, and pyruvate kinase, option C, phosphofructokinase 2 and fructose 1,6 bisphosphatase, and option D, pyruvate carboxylase and phosphoenol pyruvate carboxykinase. The reciprocal regulation of glycolysis and gluconeogenesis is mainly controlled by interplay of two key enzymes. What are these enzymes? Option A, hexokinase and glucose 6 phosphatase. Option B, phosphofructokinase 1 and pyruvate kinase. Option C, phosphofructokinase 2 and fructose 1,6 bisphosphatase. And option D, pyruvate carboxylase and phosphoenol pyruvate carboxykinase. So both of you have written option C, phosphofructokinase 2 and fructose 1,6 bisphosphatase. Okay, that is the correct answer. So these two enzymes basically, so phosphofructokinase 1 is the enzyme that regulates the conversion of, that catalyzes the conversion of fructose 6 phosphate to fructose 1,6 bisphosphate. And the reverse reaction that is required in gluconeogenesis, we have fructose 1,6 bisphosphatase that converts fructose 1,6 bisphosphate to fructose 6 phosphate. Okay, so here you can see fructose 1,6 bisphosphate will go into glycolysis and fructose 6 phosphate will be towards the gluconeogenesis pathway, right? So here there are certain energy molecules. So if you have enough ATP, you don't need you don't need more and more glycolysis. In that case, gluconeogenesis will be favored. So it will inhibit the PFK1, and it will kind of activate the FBPase1 so that the pathway goes towards gluconeogenesis. Similarly, if citrate is there, that means TCA cycle is already replenished, so it will also inhibit the glycolysis. It will favor the gluconeogenesis. If energy demanding molecules are present like ADP and AMP, that means uh, more ATP and more ATP requirement is there. So for in that case, PFK1 will be activated and FBPase will be inhibited, and the pathway will go towards glycolysis, and in that case gluconeogenesis will be inhibited. Okay, but now these two enzymes, phosphofructokinase 1 and FBPase1, they are also activated and inhibited by a molecule called fructose 2,6 bisphosphate. So this is fructose 1,6 bisphosphate. We have another molecule, that is fructose 2,6 bisphosphate, which is isomer. So in any organism, we have certain isozymes of the enzyme. So one enzyme can be present in multiple copies, or and these copies are basically there to perform different functions. So phosphofructokinase phosphofructokinase, here you can see it is written phosphofructokinase 1, right? So that PFK1 is one of the isozyme of PFK. There's another isozyme of PFK, that is PFK2, and it basically catalyzes the reaction of fructose 6 phosphate to fructose 2,6 bisphosphate. So you see the substrate for both the enzymes are same. PFK1 and PFK2, the substrate is fructose 6 phosphate, and both of the enzyme are basically transferring one phosphate group from ATP to the fructose 6 phosphate, okay, to the substrate. But you see the position of phosphate. So PFK1 is transferring the phosphate group to the first carbon of the fructose 6 phosphate. So this becomes fructose 1,6 bisphosphate, whereas PFK2, it is transferring the phosphate to the second carbon, making it fructose 2,6 bisphosphate. Okay, so in the sixth carbon already the phosphate is there. Either the phosphate is added in the second carbon, or if it is added in the first carbon, depending on that, we get fructose 1,6 bisphosphate and fructose 2,6 bisphosphate. So PFK1 is the enzyme that is there in the glycolysis, but there is another enzyme called PFK2, which converts the fructose 6 phosphate into fructose 2,6 bisphosphate. And this fructose 2,6 bisphosphate also gets dephosphorylated by an isozyme of FBPase. So here we had FBPase 1 that was converting fructose 1,6 bisphosphate. We have FBPase1 that was converting fructose 1,6 bisphosphate into fructose 6 phosphate, dephosphorylation of fructose 1,6 bisphosphate into fructose 6 phosphate. The isozyme of this enzyme, that is FBPase2, it uh, does the dephosphorylation of fructose 2,6 bisphosphate into fructose 6 phosphate. Okay, so both of them again are doing the same reaction, but at a different position. So both of them are doing dephosphorylation. One is doing the dephosphorylation of fructose 1,6 bisphosphate. Second one is doing the dephosphorylation of fructose 2,6 bisphosphate. Okay. Now this PFK2 and FBPase2, it is a bifunctional enzyme. So it is one enzyme that has both the function, both kinase and phosphatase. Okay, and how this fructose 2,6 bisphosphate inhibits or activates PFK1 and FBPase1, that we'll see. So this is this bifunctional enzyme. So PFK2 and FBPase2. So when glucagon is high, in that case, glucagon triggers the CAMP pathway, and that CAMP pathway will activate a protein kinase. Once that protein kinase is activated, this bifunctional enzyme gets phosphorylated. It is a kinase, so again, phosphorylation is the reaction that will be catalyzed. So this bifunctional enzyme gets phosphorylated. Once it gets phosphorylated, the FBPase2 becomes active and PFK2 becomes inactive in the phosphorylated version. Since FBPase2 is active, that means fructose 2,6 bisphosphate will get converted into fructose 6 phosphate, right? This is the FBPase2 reaction. So once FBPase2 is active, the levels of fructose 2,6 bisphosphate will go down. That uh, in level the level of fructose 2,6 bisphosphate going down will basically inhibit the glycolysis and stimulates gluconeogenesis. Okay, why? Because fructose 2,6 bisphosphate inhibits the FBPase1 and activates the PFK1. Okay, so low level of fructose 2,6 bisphosphate, that means more and more gluconeogenesis can happen now, because the uh, FBPase1 will not be inhibited. Okay, and once this happens, then gluconeogenesis will keep on occurring and glucose levels will increase, and after a point insulin secretion will start. Insulin secretion, what will it do? It will trigger the dephosphorylation of this bifunctional enzyme. Okay, so right now this bifunctional enzyme is phosphorylated. So once insulin is secreted, insulin will activate a phosphoprotein phosphatase, which will dephosphorylate this bifunctional enzyme. And once the dephosphorylated form is present, in that form PFK2 is active. Okay, so phosphorylated version has FBPase2 active, the dephosphorylated version has PFK2 active. Okay, now once PFK2 is active, what will happen? PFK2 active means it can convert fructose 6 phosphate into fructose 2,6 bisphosphate. This fructose 2,6 bisphosphate can stimulate glycolysis and inhibit gluconeogenesis. Why? Because fructose 2,6 bisphosphate will activate the PFK1, and that PFK1 will convert more fructose 6 phosphate into fructose 1,6 bisphosphate. That will go make the pathway go towards glycolysis, and it will inhibit the FBPase1 so that the pathway does not go towards gluconeogenesis. So presence of glucagon, as we had discussed in the previous question, presence of glucagon stimulates gluconeogenesis. How? Because when glucagon is present, it triggers the CAMP pathway, which triggers a protein kinase that can phosphorylate the bifunctional PFK2 FBPase2 enzyme. Phosphorylated PFK2 FBPase2 enzyme has the FBPase2 form as the active form. Once FBPase2 is active, it will trigger the dephosphorylation of fructose 2,6 bisphosphate, that will lead to low concentration of fructose 2,6 bisphosphate and higher concentration of fructose 6 phosphate. Now naturally fructose 6 phosphate is high, that means it can go towards gluconeogenesis. Low levels of fructose 2,6 bisphosphate will inhibit the glycolysis and stimulate gluconeogenesis. Okay, because it will basically inhibit the PFK1 enzyme and activate the fructose 1,6 bisphosphatase enzyme. Okay, once insulin is secreted, in that case phosphoprotein phosphatase is activated, and then again dephosphorylation of this bifunctional enzyme occurs. Dephosphorylation leads to PFK2 activation, and that active PFK2 can now synthesize more and more fructose 2,6 bisphosphate that can stimulate glycolysis and that can inhibit gluconeogenesis. Okay, is this part clear? So how PFK2 and FBPase2 are basically regulating the activation and inhibition of glycolysis and gluconeogenesis as and when required. So if the body requires more glucose, gluconeogenesis will be triggered. If the body requires more energy, glycolysis will be triggered. Okay. So depending on what the body needs, these hormones that are glucagon and insulin, those will be secreted, and the activation and inactivation of PFK2, FBPS2 will occur, which will in turn activate or inactivate the gluconeogenesis or glycolysis pathway depending on the hormone that is secreted. Is that clear? If any of you have any doubt, please let me know. Okay, then I'll move on to the next question. The Cori cycle refers to the process of converting lactate produced in muscle tissue back into which molecule in the liver? The Cori cycle refers to the process of converting lactate produced in a muscle back into which molecule in the liver? Option A glucose, option B pyruvate, option C alanine, option D acetyl coenzyme A. The Cori cycle refers to the process of converting lactate produced in the muscle tissue back into which molecule in the liver? Option A glucose, option B pyruvate, option C alanine, and option D acetyl coenzyme A. Okay, both of you have written option A glucose. Okay, so all of you are agreeing to option A glucose. That is the correct answer. So Cori cycle basically uh, is the conversion of lactate into glucose, lactate back to glucose. So basically what happens is when you do vigorous exercise, you have observed that there's a muscle pain that happens after running very fast or having some kind of very vigorous exercise. So that why that happens is due to the production of lactic acid or lactate. So what happens is when our body needs energy, performs glycolysis. So it continuously converts glucose into pyruvate, and then pyruvate pyruvate gets converted into acetyl coenzyme A, entering the TCA cycle and ultimately generating ATP. But in case our body cannot meet the requirement of glucose, what it does, instead of converting the pyruvate into acetyl coenzyme A, the pyruvate gets converted into lactate in the absence of oxygen. So in the presence of oxygen, we have oxidative phosphorylation that occurs, but in the absence of oxygen, pyruvate gets converted into lactate, and that pyruvate that lactate keeps getting accumulated in the muscle, causing the pain. and higher level of lactate accumulation can actually cause lactate acidosis also. Okay, but to prevent uh, to prevent acidosis formation, what our body does, it has a very regulated mechanism that when lactate accumulation occurs in the muscle tissues, it immediately transports the lactate into the liver and converts it back to glucose. So it will convert it back to pyruvate and then to glucose. So here you can see when during anaerobic glycolysis, or in the absence of oxygen, when you're doing exercise, the oxygen levels are not uh, in coordination with the body in the with the requirement by the body. Okay, so in that case what happens is not all glucose molecules that is getting converted into pyruvate can enter the oxidative phosphorylation, because oxygen is not there as the terminal electron acceptor. In that case, pyruvate gets converted into lactate anaerobically in the muscles. Okay, now this lactate keeps getting accumulated in the muscle, causing uh, lactate acidosis. But to prevent that, our body has a mechanism where this lactate is transported out of the muscle into the blood and then again transported into the liver. And this liver, what it will do, it will convert this lactate back to pyruvate, and pyruvate will again get converted into glucose 6 phosphate and then get converted into glucose, and that glucose will be transported back to blood. Okay, so in erythrocytes and during anaerobic glycolysis, skeletal muscles, pyruvate is reduced to lactate by lactate dehydrogenase, and this lactate is then converted back to glucose in the liver by and this process is the Cori cycle. Okay, so this reaction serves two critical functions during the anaerobic glycolysis. It yields NAD plus, and uh, it also, so when lactate dehydrogenase converts pyruvate to lactate, it yields one NAD plus molecule, and then this lactate gets converted back to glucose, that is the Cori cycle. Okay, I hope that's clear to everybody. Any doubt in this? Okay, then I can move on to the next question. Which coenzyme is involved in the transfer of reducing equivalents during the oxidative phase of HMP shunt? Which coenzyme in the transfer of reducing equivalents during the oxidative phase of HMP shunt? Option A NADH, option B NADPH, option C FADH2, and option D coenzyme A. Which coenzyme is involved in the transfer of reducing equivalents during the oxidative phase of HMP shunt? NADH, NADPH, FADH2, and coenzyme A. So I can see all of you have written option B, that is NADPH, as the answer. That is the correct answer. So HMP shunt pathway is basically a pathway to produce to synthesize NADPH and pentose sugar. Okay, so it's an alternative to glycolysis and TCA cycle, but only around 10% of glucose enters into this HMP shunt pathway. Rest most of the glucose is metabolized using glycolysis and TCA. HMP shunt pathway is the main source of NADPH in our body. Uh, Almost 95% of NADPH is uh, produced by HMP shunt pathway. This pathway starts from glucose, and then it basically creates 5 sugar molecules. So that's why pentose phosphate pathway. It is also called pentose phosphate pathway. So it creates pentose sugars, and it synthesizes NADPH. So synthesis of NADPH and pentose sugar is the main uh, like main reason why this pathway occurs in our body. Okay, so NADPH is the correct answer. Moving on to the next question now. In the oxidative phase of HMP shunt, which enzyme converts glucose 6 phosphate to 6 phosphoglucono-D-lactone? In the oxidative phase of HMP shunt, which enzyme converts glucose 6 phosphate to 6 phosphoglucono-D-lactone? Option A, glucose 6 phosphate dehydrogenase. Option B, trans ketolase. Option C, trans and aldolase. And option D, 6 phosphogluconate dehydrogenase. In the oxidative phase of HMP shunt, which enzyme converts glucose 6 phosphate to 6 phosphoglucono D-lactone? Option A, glucose 6 phosphate dehydrogenase. Option B, trans ketolase. Option C, trans aldolase. And option D, 6 phosphoglucono dehydrogenase. So I can see two of you have written option A, that is glucose 6 phosphate dehydrogenase. Glucose 6 phosphate dehydrogenase converting glucose 6 phosphate to 6 phosphoglucono D-lactone. That is the correct answer. So this is the oxidative phase of HMP shunt pathway. So I told you it starts with glucose 6 phosphate. So glucose getting converted into glucose 6 phosphate. After that, glucose 6 phosphate in glycolysis goes to fructose 6 phosphate, right? But in HMP shunt pathway, it will be oxidized to 6 phosphogluconolactone by an enzyme glucose 6 phosphate dehydrogenase. Now, if you remember in the previous class also, I have told you wherever dehydrogenase enzyme is present, you will have either NADH reduction or NADPH reduction. Okay, so sorry, basically NAD reduction or NADP reduction. So here NADP is reduced to form NADPH, and in turn glucose 6 phosphate is oxidized to form 6 phosphogluconolactone. So the whole, even if you don't remember the structure, just keep looking at the structure. So this is the 6 carbon sugar, and here instead of the alcohol group, it gets converted into a ketone group. So it's a lactone, uh, so it's a cyclic molecule actually, and it's a lactone, so gluconolactone. Okay, so 6 phospho, because the 6th carbon has the phosphate attached to it, gluconolactone. Okay, so glucose 6 phosphate gets converted into 6 phosphogluconolactone. by the enzyme glucose 6-phosphate dehydrogenase, converting one NADP molecule into NADPH. So this is one first source of NADPH. Then this 6-phosphogluconolactone gets hydrolyzed by the hydrolase enzyme, gets hydrolyzed, that is addition of H2O occurs. So you can see here where the addition of H2O is occurring, the ring opening will happen. So here one OH is, so here we have O, so one H is added here, and then one OH will be added here. So you can see O minus. So H2O plus is added, and you have one OH here, and we have one H added here. Okay. So hydrolase does the hydrolysis reaction, addition of H2O breaks the ring, and it forms 6-phosphogluconic acid. So from the ketone we have a COH group, that is the acid group. Okay. So started with glucose 6-phosphate, it got converted into 6-phosphogluconolactone by glucose 6-phosphate dehydrogenase. Then we have hydrolase that will break the lactone ring and form 6-phosphogluconic acid. This 6-phosphogluconic acid now will again get oxidized into a 5 carbon sugar, that is ribulose 5-phosphate. So 6-phosphogluconic acid by the enzyme 6-phosphogluconate dehydrogenase get converted into ribulose 5-phosphate, and this is also, I told you, this is a dehydrogenase enzyme, so that means one NADP molecule will be converted into NADPH. So this is the second NADPH. So this is called the oxidative phase because two oxidation steps are occurring. First is here where glucose 6-phosphate is getting converted into 6-phosphogluconolactone, and then 6-phosphogluconic acid is getting converted into ribulose 5-phosphate by enzyme 6-phosphogluconate dehydrogenase. So in both the steps, reduction of NADP plus is occurring and NADPH is forming. So NADPH formation, this reduction is in turn oxidizing the substrate glucose 6-phosphate and 6-phosphogluconic acid. Okay. So 6-phosphogluconate dehydrogenase, it is along with oxidizing, it is also eliminating one CO2 molecule from the this 6 carbon sugar. So 6-phosphogluconic acid, you can see there are 6 carbons, 1 2 3 4 5 6. So one CO2 is released, and now we have a 5 carbon sugar molecule, that is ribulose 5-phosphate. Okay. So 1 2 3 4 5 5 carbons are there. Okay. So one carbon is eliminated. So this is the first part of the HMP shunt pathway where a pentose sugar and 2 NADPH molecules are synthesized. Then this ribulose 5-phosphate further gets metabolized into different sugar molecules. Okay, that we'll see in the next question. So oxidation of 3 molecules of glucose by pentose phosphate pathway or HMP shunt pathway results in the production of dash. Option A, 3 molecules of pentose, 6 molecules of NADPH and 3 molecules of CO2. Option B, 4 molecules of pentose, 6 molecules of NADPH and 3 molecules of CO2. Option C, 4 molecules of pentose, 3 molecules of NADPH and 3 molecules of CO2. And option D, 3 molecules of pentose, 4 molecules of NADPH and 3 molecules of CO2. Oxidation of 3 molecules of glucose by pentose phosphate pathway results in the production of dash. 3 molecules of pentose plus 6 molecules of NADPH plus 3 molecules of CO2. Then we have option B, 4 molecules of pentose plus 6 molecules of NADPH plus 3 molecules of CO2. Option C is 4 molecules of pentose, 3 molecules of NADPH, and 3 molecules of CO2. And option D, 3 molecules of pentose, 4 molecules of NADPH, and 3 molecules of CO2. So we have two answers. Both of them are saying option A. 3 molecules of pentose, 6 NADPH, and 3 CO2. So from 3 glucose, how many pentose and how many CO2 will be released? So you can solve this question logically also. I told you HMP shunt pathway is uh, a pathway to synthesize NADPH and pentose sugar. So glucose is a 6 carbon molecule, right? So from glucose molecule, you saw in the oxidative step itself, one carbon dioxide is released to form a 5 carbon pentose sugar, right? So now if 3 molecules of glucose are there, then that means how many carbons are there? 6 carbon and 1 glucose. So 3 molecules of glucose means 18 carbons, right? So we have 18 carbon. And you saw in the oxidative step, in this case, 1 molecule of glucose is leading to 2 molecules of NADPH. Okay, so that means 3 molecules of glucose will give 6 molecules of NADPH. Okay, so now we have 18 carbon. From those 18 carbon, first every molecule of glucose will release one CO2. So 3 molecules of glucose are going to give 3 CO2. So 3 carbons are uh, gone. So remaining how many carbons? 18 minus 3 is 15, right? And now 15 carbons and we need pentose sugars. So that means 5 carbon in each sugar. So 15 by 5 is 3. So that means we have 3 molecules of pentose, 3 molecules of CO2. So if you calculate the number of carbons, 3 molecules of pentose, that means 5 3s are 15. 15 plus 3 molecules of CO2, that is 15 plus 3, 18. So 3 molecules of glucose are leading to 3 molecules of pentose and 3 molecules of CO2. Along with that, double the number of NADPH, because from 1 molecule of glucose, we are getting 2 molecules of NADPH. Okay, so 3 molecules of glucose can give 6 molecules of NADPH. So the correct answer is option A. So this is the HMP shunt pathway. So I told you so this is just an example where if we have 6 molecules of glucose 6 phosphate, so we are starting the pathway from glucose 6 phosphate. Glucose 6 phosphate gets converted into 6 phosphogluconolactone by glucose 6 phosphate dehydrogenase that we saw in the oxidative step, where we get one molecule of NADPH per molecule of glucose 6 phosphate. Okay, so if we have 6 glucose 6 phosphate, we will get 6 NADPH and 6 molecules of 6 phosphogluconolactone. This 6 phosphogluconolactone will get converted into 6 phosphogluconic acid, that by the enzyme gluconolactone hydrolase. The 6 phosphogluconic acid or 6 phosphogluconate will then again converted into ribulose 5 phosphate by the enzyme 6 phosphogluconate dehydrogenase. This enzyme, if you see, this enzyme again requires NADP molecule and converts it into NADPH. So one NADPH we are getting from this step, and one NADPH we are getting from this step. Okay, so 6 molecules of glucose 6 phosphate is yielding us 12 molecules of NADPH in total. 6 from here, 6 from here. Okay, and one carbon dioxide is related from each glucose 6 phosphate molecule to become ribulose 5 phosphate, because this is a 6 carbon molecule. So this is also 6 carbon molecule, 6 phosphogluconic acid. This is a 6 carbon molecule. So from each molecule of 6 phosphogluconic acid, one carbon dioxide is eliminated to form ribulose 5 phosphate. So 6 molecules of phosphogluconate can give 6 molecules of ribulose 5-phosphate. Okay, till this point, this is the oxidative step of HMP shunt pathway. Okay, then we have an isomerization reaction. Isomerization basically means the molecular formula is same, the number of carbon, hydrogen, oxygen will be same, only the structure will be different. So 6 molecules of ribulose 5-phosphate will yield 4 molecules of xylulose 5-phosphate and 2 molecules of ribose 5-phosphate. So this ribulose 5-phosphate, by an isomerase enzyme, it will get converted into a ribose 5-phosphate, and by epimerase enzyme it will get converted into xylulose 5-phosphate. Okay, so both of these are 5 carbon uh, sugars. Okay, so but amount of xylulose 5-phosphate is higher than ribose 5-phosphate. Now there's a transketolase reaction where 2 molecules of xylulose 5-phosphate will uh, along with 2 molecules of ribose 5-phosphate, transketolation will occur, and now you have 5 molecules here, 5 carbon atoms here and 5 carbon atoms here, right? Total number of carbon atom is 10. So we'll get two sugars. One is a 7 carbon sugar and one will be 3 carbon sugar. So we had xylulose 5-phosphate and ribose 5-phosphate. From each molecule of ribose 5-phosphate and xylulose 5-phosphate, we will get one pseudoheptulose for 7-phosphate, that is a 7 carbon sugar, and one glyceraldehyde 3-phosphate, that is a 3 carbon sugar. Okay, so total number of carbon remains same, it's just a transketolation that is happening. Okay, then next is a transaldolase reaction where the glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate, these two uh, combine and become 2 molecules of fructose 6-phosphate and 2 molecules of erythrose 4-phosphate. So this is 3 carbon and this is 7 carbon, total 10. Now we are getting one 6 carbon molecule and one 4 carbon molecule. So from 10 again we have 10 carbons total. So number of carbon remains the same, only the type of sugar is changing. It from a 7 carbon sugar, now we have 6 carbon sugar and a 4 carbon sugar. Okay, so we have erythrose 4-phosphate and fructose 6-phosphate. Now this fructose 6-phosphate can get converted into fructose 1,6 bisphosphate, and basically it can enter both glycolysis and gluconeogenesis. And the erythrose 4-phosphate that we had, that will convert again have a second transketolation reaction by a second transketolase enzyme. So you remember we had 4 molecules of xylulose 5-phosphate. So those 4 molecules of xylulose 5-phosphate, 2 were involved in the first transketolation where we had pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate. Now these two, the 2 remaining xylulose 5-phosphate, they will do the second transketolase reaction with the erythrose 4-phosphate. Now this is a 4 carbon and this is 5 carbon. So how many carbons are there? 9. Okay, so we will get 2 molecules of fructose 6-phosphate, that is 6 carbon, and remaining are 3 carbon. So we will get glyceraldehyde 3-phosphate. Okay, so this is the second transketolase. This fructose 6-phosphate will also go back to glucose 6-phosphate, and then the glyceraldehyde 3-phosphate will also get converted into fructose 6-phosphate. So 2 molecules of glyceraldehyde 3-phosphate, that is 2 3 carbon sugars, will in total give 1 6 carbon sugar, that is 1 molecule of fructose 6-phosphate. So how many molecules total are there? 2 plus 2 plus 1, that is 5. Okay, so 5 molecules of fructose 6-phosphate will go back to glucose 6-phosphate, that is the reversal of glycolysis. So here 5 glucose are regenerated, 6 CO2 are eliminated, 12 NADPH are generated and 1 glucose is completely oxidized. What do you mean by complete oxidation? Complete oxidation means conversion of any organic molecule into CO2. Okay, that is the definition of complete oxidation of organic molecule. Any organic molecule like glucose is completely getting converted to CO2. So we started with 6 glucose 6 phosphate and we ended up with 5 glucose 6 phosphate, and 1 glucose 6 phosphate got converted into carbon dioxide completely. So complete oxidation. So 1 glucose 6 phosphate got converted into 6 CO2. That is, all the carbons got oxidized and the complete breakdown of that glucose 6 phosphate occurred. Okay. So this is the total reaction of HMP shunt pathway, where glucose 6 phosphate with along with two NADP molecule and in presence of water gets converted into ribose 5 phosphate, that is a pentose sugar. I told you the uh, outcome of this pathway is a pentose sugar and a NADPH molecule. So we get from one molecule of glucose 6 phosphate we get one pentose sugar, one carbon dioxide, because one of the molecule gets completely uh, oxidized, and two molecules of NADPH and two molecules of protons. Okay, so this is the total reaction, complete reaction of HMP shunt pathway. Is this clear to everyone? So this is the last question of the session. So let me know if you have any doubt in any of the pathways. So we have discussed glycolysis, we have discussed TCA, we have discussed the regulation of TCA, we have discussed glycolysis and gluconeogenesis, how they are related, what are the differences, which particular steps are different in glycolysis and gluconeogenesis. So there are major three uh, steps where the differences are there. Rest all the enzymes remain same. Those three irreversible steps of glycolysis are uh, performed by different enzymes in the reverse direction. In case of gluconeogenesis, we have discussed the regulation of those enzymes, and then we saw Cori cycle, and so there is another part that we haven't discussed is the feeder pathway of glycolysis. So in the glycolysis, along with glucose, we have other molecules that feed the glycolysis at certain steps. Like, for example, sucrose can break down into glucose and fructose. So glucose can enter the glycolysis directly, whereas fructose can directly enter the glycolysis uh, in the step of fructose 6 phosphate. So fructose can get converted into fructose 6 phosphate and then enter the glycolysis. So similarly, there are other molecules. So that it is already given in your slides, so you can refer to that. Then we discuss the Cori cycle. We have then we move to HMP shunt pathway, which I explained you right now. So if you have any doubt in any of the pathways and any of the regulation, how it is happening, please ask. Then we can have a discussion on that. Do you have any doubt? Any difficulty in understanding any of the pathways, please let me know. Is it clear then? So basically, you don't have to mug up these pathways. You can you should try to understand the pathway, how it is proceeding, and automatically you will see that you will be able to write the pathway without actually mugging it up. Okay, so if you understand what is happening, how many carbons we are starting with, how many carbons we are ending up with, so you can logically write the pathway. Learning the structure is not necessary, I would say, and it's not really possible to remember the structure of all the molecules. But once you understand the pathway, you will automatically see that you will understand the structure also. So you will be able to draw the structure as well. So how the reaction, each and every step is proceeding, just pay attention to that and try to learn it. So can I end this session if there's no doubt? You can write it in the chat box if you have any question. We have time in this session, so we can discuss. If there are no more questions, then we can end this session. Okay. Okay, then. Thank you all for joining. I'll close the session now.